right, we'll get started now. Thank you all for being here. I'm Olivia, and I'm really excited to be introducing our talk today about There's More to the Messenger, the Wonderful World of RNA. We are Science in the News from Yale Science Communication, which is an organization of graduate students at Yale committed to engaging and informative science outreach. You'll be hearing from 3 speakers today about the different types of RNA that can either help or hinder us. Each speaker will have 15 minutes to speak, and we'll have time for questions at the very end. Please feel free to type your questions in the Q&A as we go, and we'll collect them all at the end to answer them. So here are our speakers today. First, we're going to hear from Brahmmi in the chemistry department, and she studies the dynamics of protein and RNA interaction in the spliceosome. And you'll know a little bit more about what all those terms mean after her talk. And her fun fact is that she loves listening to podcasts. Next, we have Mikey in the Department of Microbial Pathogenesis, and he studies SARS-CoV-2 structure and pathogenesis and has an extremely pink guitar. And finally, we'll hear from Matt in the Department of Molecular Biophysics and Biochemistry, where he studies the misfolding of tumor suppressor proteins in cancer, and in his spare time loves playing soccer. So before I pass things off to the speakers today, I want to give you a little introduction about what you're going to be hearing about. So from the title of the talk, you can intuit that we're going to be talking about RNA, but why should we care about RNA? Well, RNA is a small molecule, but it can affect many different aspects of our life. So we're going to learn about many different diseases that might have their roots in defects in RNA, as well as virally transmitted diseases that might be spread by mosquitoes that also rely on RNA. But on the good end of things, we'll learn how RNA goes into vaccines like the recent Moderna and Pfizer COVID vaccines that many of us have been so happy to have these last few years. So what is RNA? To talk about RNA, first we have to talk about DNA. And DNA is the instruction manual that contains all the information needed to make you yourself. So we can envision the DNA as a huge book full of pages upon pages of instructions. And these instructions, just like the instructions we would have in words, are made up of individual letters strung together. This instruction booklet has to be converted using these different sets of instructions into proteins. These are the molecular machines that basically are responsible for all of the functions in your body. So we have to translate these instructions into machines. And the way we do that is with RNA. We call this the central dogma of biology, that DNA makes RNA makes protein. This is the link between the instructions and the product. So DNA and RNA are similar in that they're both made of nucleotides, which are these letters that I was talking about before. And these specific letters make it so that the DNA and RNA can bind to one another and to themselves, and that they can carry information in the form of these letters from one molecule to the other. They differ in that DNA is double-stranded and RNA is single-stranded. So in order to get this information from the DNA to the molecular machines, there's a 2-step process. The first step is called transcription, and this is where DNA is converted into RNA. And you can think of this as writing some letters from one kind of letter to another, just transcribing. Whereas a second step, translation, converts RNA into these proteins, these molecular machines, and that's a much different process. So translating from these letters into these objects here. So we're going to pass it off now to Brahmmi first, who's going to talk to us about a tiny molecule with big impacts, the science behind how RNA in your cells is processed to make us who we are, and how this can go wrong, where even a tiny mistake in this process can lead to drastic consequences. Next, sort of following this RNA molecule here, we'll hear from Mikey, who will take us on a historical tour of how RNA can be used against us by viruses like yellow fever. He'll explain to us the massive impact that these viral RNAs can have on human health, as well as explaining the gritty details of how the scientists of the past made discoveries that still impact us today. And finally, we'll hear from Matt, who will open us to an even wider world of RNA by explaining how these tiny messengers can be folded like origami to become tools of all kinds. He'll tell us about how your cells are using these tools naturally right now, as well as how scientists hope to harness them for the technologies of the future. And with that, I will stop sharing my screen and pass things over to Brahmmi to start us off.
Thank you, Olivia, for that lovely introduction. And thank you guys for being here and joining us for this series of RNA talks. So today I'm going to be talking to you about messenger RNA and how it's associated with a very specific disease and how processing of messenger RNA can result in some severe consequences. So we've all heard about the double-stranded DNA and how it has hugely impacted the field of science and how it has resulted in a lot of discoveries after that. But what's more important here for us today is RNA and how RNA has definitely been central to a lot of molecular biology processes and how it is used now recently in several different RNA technologies that you'll hear about later on in the talk. So today I want to refer back to this every once in a while, the central dogma of molecular biology. So we go from a double-stranded DNA, which has the instructions to make the protein. Then you have RNA, which is produced by the process of transcription. And now this is messenger RNA. There are several other types of RNA, and you'll hear about that later on. But we're referring to this as messenger RNA. It carries the messages that are housed in the DNA to then finally make protein in the process of translation. However, at this step, there's a lot of processing that occurs for the production of messenger RNA, and it can have several big consequences if things go awry and we run into errors. And so such is the case in Hutchinson-Gilford progeria syndrome, where we have this genetic abnormality that is characterized by mistakes in messenger RNA processing, and it is associated with dramatic and rapid appearance of aging. So you're going to see like loss of subcutaneous fat, you're going to see hair loss. The average lifespan is actually about 14 years, and it is a genetic condition. You have higher risk of heart disease and stroke, and this is known as progeria or HGPS. And so this specific disease is related to one tiny process of this messenger RNA being made, and I'm going to be telling you about that today. So if we kind of go back to the central dogma of molecular biology, we have DNA that gets turned into messenger RNA through the process of transcription, and that's the step that we will be focusing on today for the sake of this talk. So think about DNA, which houses genetic information that then gets processed into messenger RNA. Now, this messenger RNA still houses all of that information that you get from the DNA molecule. However, you have these like protein coding regions within the messenger RNAs. So you imagine a long, big strand of information, and the strand of information has protein coding regions known as exons. So these are letters that represent that there is information within the strand. Then you have messenger RNA. Again, these are the letters that I'm referring to, that then finally get translated into proteins. So that is our final goal. And proteins are central to proper functioning of cells, and that is primarily the reason why we're still alive, because proteins are needed for everything in our body. So our end goal is the protein, But for production of protein, we need messenger RNA. And that is the processing step that is required for every protein production. So what's interesting is that when we have messenger RNA being formed, we have this thing that is the tiny step before it that has pre-mRNA. So DNA doesn't just get transformed into messenger RNA. It gets first produced into pre-mRNA, and it actually has some unnecessary information. So if we kind of just look at this, these color-coded regions are known as the protein coding regions that I just referred to in the previous slide. And these are the exons. These are necessary for the production of protein. What is not necessary is the black regions right there. Those are the unnecessary regions, and they need to be, they are part of the pre-mRNA once the DNA undergoes transcription. So now I've color-coded the blue, the pink, and the green over here and referring them to as exon 1, 2, and 3 as the protein coding regions. The black regions, which are the unnecessary information regions, are known as introns. And so we remove the introns and you have processing that results in the ligation or putting together the exons. And then after translation, you get the formation of proteins. So it just kind of makes sense that unnecessary information must be removed when we need to kind of get to this end product with only exons and no introns. And this happens by the process known as RNA splicing. So what does that mean? Well, if we take a look here, DNA carries all the genetic information, including the necessary protein coding regions and the unnecessary non-protein coding regions known as introns. All of that gets transferred into the RNA, and by the process of RNA splicing, you have ligation of the protein coding regions and removal of the non-protein coding regions known as introns. And after that, we have this final messenger RNA strand that is going to result in the production of proteins. What's really cool about eukaryotic mechanisms is that we actually use this thing called alternative splicing to increase protein diversity. And by that, I mean, imagine a strand of RNA with protein coding regions that are colored here, 1 through 5. The black line represents the introns, the non-protein coding regions that are not necessary information. With alternative splicing, we can actually generate different combinations of protein coding regions to form different proteins from the same RNA molecule. And this is very unique to humans and other eukaryotic organisms. And it's the cause of protein diversity. So you're getting 3 different proteins from the same RNA strand. And this is very complex, and it generates messenger RNA that then results in production of 3 different proteins. So this process is actually done by a molecular machine known as the spliceosome. So it's this really big machine with like over 200 molecules involved in it. And what it does is that it recognizes the spot where it needs to cut out the unnecessary information. It needs to first figure out, where do I go, and cut out the unnecessary information, and then put together the necessary information, the exons. So it jumps on, binds to the intron and exon sites, and then carries out this process known as RNA splicing. And so basically at this is very important in order for messenger RNAs to be processed correctly. However, at this point, the possibilities for problems are endless. So you can imagine some of the ways in which processing of messenger RNA can go very wrong. For example, what would happen if we have a mutation or an error in the spliceosome itself. Let's say some of the molecules are missing in the spliceosome. Therefore, the spliceosome may not be able to function correctly. Or, for example, what if it doesn't find the right region to bind? If it doesn't recognize the correct location to bind, it's not going to bind there, and it will result in errors downstream. What about mutations in the protein coding region? What happens if you have a change in what the protein coding region looks like? Well, you're not going to form the right type of protein, and that's going to result in a completely different function, and the protein is not going to behave the way it should. So again, the possibilities for problems are endless. And if we go back to Hutchinson-Progeria-Gilford syndrome, I will talk to you about how RNA processing is involved in one of those steps. So just really quickly though, I want to share what splice sites are. Spliceosome needs to find the right location to remove unnecessary information. So I've told you that the spliceosome needs to locate a very specific sites in the pre-mRNA molecule. So it's going to refer to be as the 5' splice site and the 3' splice site. And it's going to attach to those regions in order to carry out splicing. So in the case of Hutchinson-Gilford progeria syndrome, what ends up happening is you have a normal 5' splice site where the spliceosome should bind. However, a mutation causes a switch in the 5' splice site. So instead of using this site, it uses this site. And that results in deletion of this region of exon 11, which is the protein coding region. So then now you have a shortened or truncated exon 11, and that results in a mutant protein being formed. So sure, these are ligated together and processing has occurred, but now you have a mutant protein that doesn't do the same function as the normal protein. So you have exon 11, which results in the formation of progerin, which is the mutant protein. You have the cryptic 5' splice site, so not where it's supposed to cut, and that results in the truncated exon 11. In the DNA sequence, if we kind of take a closer look here, you can see that there is, in the genetic information, a single nucleotide mutation. So there's a C to T substitution in the DNA strand itself that then results in massive errors in processing that results in HGPS. And that then goes back to the wrong 5' splice site selected, resulting in the production of progerin. And so what are the consequences here? Well, if we kind of look at the normal side of things, we have production of the normal protein, the pre-lamin A protein, which then gets processed and it results in the production of lamin A protein. Now, lamin A protein is very, very important for the structural integrity of cells. So it gives the cells sturdiness, and it is important for the nuclear envelope of cells. So in that, the normal protein results in healthy cells. However, with the mutant protein, now you have the production of progerin, and it is not doing the same thing that we would expect the normal protein, the lamin A protein, to do. So you have processing of, excuse me, I forgot to type this out, but you now have processing of progerin that is not happening, and that means you don't have lamin A protein being made. And that results in accumulation of progerin, which results in severe problems, which are now apparent in HGPS. So from here, in this talk, I've shared that problems in the cell can have long-term impacts. The lack of lamin A protein can actually result in downstream effects that we can see on the outside as a result of a single nucleotide substitution and errors in messenger RNA processing. So that is the core reason why we have Hutchinson-Gilford progeria syndrome. There are treatments that scientists are working on. However, we haven't finalized any such treatment so far. And in conclusion, RNA alternative splicing can dictate several splicing outcomes, but it is also one of the very steps where problems can occur. HGPS is caused by a single nucleotide mutation, which results in early aging and other health issues. So in this disease, you have the spliceosome that recognizes the wrong splice site, which results in a lot of problems. And that RNA processing overall is important in protein production. And without RNA processing, we would not be here the way we are right now. And with that, I have shared with you several different ways in which RNA processing is important to biological functions. And now Mikey is going to share how RNA plays a role in the yellow fever virus. Thank you.
Thank you, Brahmmi. That was very interesting. Okay, so hi, everyone. I'm excited to be presenting this to you today. So while Brahmmi talked to you about how RNA is essential for cellular functions, I'm going to be talking to you about how RNA can be an intruder in the form of a virus and get inside and really mess up your cellular functions. So let's get started. So first, we'll start with what is a virus? So a virus is first composed of a nucleic acid, an intruder nucleic acid, either DNA or RNA. And in our case, we're talking about an RNA virus. And a virus is enclosed in a protein coat or capsid, which helps to protect the RNA and helps the virus attach and enter into cells. And another important thing that makes a virus is that it needs our host cells to replicate. It hijacks our cells, enters in there, makes a whole bunch of more copies of itself, and then leaves our cells left for dead and goes on to infect more cells. So one of my favorite viruses is yellow fever virus. So this is a virus that's transmitted by the mosquito, Aedes aegypti, and humans and non-human primates are the host species. So what does yellow fever look like in peoples? Well, first you have an acute phase, which looks like a very typical virus infection, possibly. You have a fever, headache, muscle aches, maybe some nausea. Then you might start to feel a little better, go into what's called a remission, but then approximately 15% of patients will progress into this toxic phase, where you can get liver and kidney failure, jaundice, such as yellowing of the skin and eyes, black bloody vomit and bleeding from places that should not be bled from, and people who progress into this phase have about a 20 to 50% mortality rate. So some death facts about yellow fever. There's still approximately 200,000 cases and 30,000 deaths each year, the vast majority of those being in Africa. An interesting fact is that in 1793, yellow fever virus was a big outbreak in Philadelphia that killed approximately 10% of its population. And yellow fever virus also killed approximately 20,000 French workers during the French attempted construction of the Panama Canal. And this Panama Canal lies here in this area where yellow fever virus is now recommended to be vaccinated against. So these areas highlighted in tan are where vaccinations are recommended. And in these areas, yellow fever is spread between and among humans and non-human primates. All right, so controlling yellow fever virus, essential to the success of the United States. We needed to get this virus under control so that we could build the Panama Canal and increase our military power and economic power. So you can see here, the Panama Canal route essentially cut the distance in half from shipping things from the US East Coast to the West Coast. So you no longer had to sail all the way around the tip of South America, say hi to the penguins in Antarctica, and come back to the West Coast. All right, and also during the Spanish-American War, they were having approximately 13 times more soldiers dying of yellow fever virus than of actual battle. So in terms of economic success and military success, we had to get this canal built. So the story I'll tell you today is a story of science and discovery, but also a story of people and people who used the tools that they had at the time to try to get this figured out. So Carlos Finlay was the first guy who proposed that yellow fever virus could be spread by mosquitoes, but nobody really believed him. That's because he probably didn't have sufficient evidence. But then enter Walter Reed in the early 1900s, and he used human experiments to more definitively show that yellow fever virus is spread by mosquitoes. Then incoming William Gorgas took this information and used it to eradicate yellow fever virus in the canal zone so we could build it successfully. And finally, Max Theiler in the 1930s created the yellow fever vaccine that we still use today. So first I'll start with the amazing work and interesting work of Walter Reed in this publication, which I highly recommend reading if you're interested. All right, so these experiments began at Camp Lazear in Cuba, and they really wanted to figure out how is yellow fever virus spreading? Is it spreading through fomites or objects likely to be carrying infectious substances, or is it spread by mosquitoes as Carlos Finlay had proposed? So here's a photo of Camp Lazear. You can see it's in this big open field looking like in Cuba, in these little huts where they did some experiments. And they paid volunteers $100 if they joined the study, and another $100 if they contracted yellow fever virus. This equates to about $3,200 today. And they gave volunteers these informed consent forms, meaning that it was a document that was explaining what was going to happen in the study, what might be some risks involved, etc. It was actually one of the first cases in biomedical research, people providing these informed consent forms. But one thing that was on the form that was interesting was that they were telling people that, hey, you're probably going to get yellow fever on this island anyway, so you might as well get it and then have us take care of you afterwards. So that was fairly interesting, actually, reading these informed consent forms that they were giving people. And they infected people with yellow fever virus using mosquitoes who had bit other people who had been infected with yellow fever. And they, along the way, had to keep careful and meticulous track of these mosquito bites and of the symptoms. So yes, they asked if yellow fever was spread by fomites or by mosquitoes. So they had these volunteers who were all so happy to be there, stay in two separate buildings, and one was called the Soiled Clothing and Bedding Building. So this had poor ventilation, windows on the same side of the house, and they filled it with clothing and bedding from yellow fever virus patients that were contaminated with blood, sweat, stools, and vomit, as contaminated as possible. And they made people stay here in 3 weeks in strict quarantine. It sounds terrible, but no person in this study had actually contracted yellow fever this way, as disgusting and unsanitary as this was. But building number 2, the infected mosquito building, this had great ventilation, clean bedding and clothing. It was basically like a 5-star hotel, but they released some mosquitoes in there which were carrying yellow fever, and lo and behold, they had a volunteer contract yellow fever. So this, along with several other experiments, really emphasized the role of the mosquito in spreading yellow fever and not necessarily fomites. When they were doing these experiments, they had to keep track of how their patients were doing over time. And so one thing they did with the tools that they had was just, check people's body temperature over time. So this is the progression of yellow fever in a patient. And you could see the spike in temperature on a few days post-infection, which then seems to get a little better, but then on the 9th day, it looks like they had a relapse, which may have been a progression into this toxic phase that I talked about earlier. Okay, so these experiments at Camp Lazear were good evidence, but they wanted more evidence, as scientists often do. You need a lot of evidence to make conclusions. So they carried out further trials at the Las Animas Hospital in Havana, so they could have more control over the number of mosquito bites and try to have better patient care afterwards. But unfortunately, when they did this, in the first study, they had 13 cases of people who got yellow fever, but nobody died. They were very lucky. However, when they did these experiments in the Las Animas Hospital, they actually had 3 people die. And one of them was a nurse. Her name was Clara Mas. It was one of the only women that participated in the study. And this is the fever chart leading up to her death. Very unfortunate. Okay, so we no longer carry out human experiments this way. We have more developed ethical guidelines for these things. And one of these ethical guidelines is the Nuremberg Code, which actually came in response to experiments that were conducted by Nazi physicians during World War II. So these Nazi physicians were put on trial, and that's where the court came up with this code for conducting human experiments called the Nuremberg Code. And one point of this code is that no experiment should be conducted where there is an a priori reason to believe that death or disabling injury will occur. And yellow fever at the time was known to be a deadly virus. So it's, arguable that Walter Reed's experiments were unethical by these standards. Okay, but one of the positive things that came out of this experiment was, of course, now we knew that we had to control the mosquitoes to control the disease, and William Gorgas took charge of this. And one of the first things he did was make hospitals mosquito-proof, so that when sick yellow fever virus patients were coming into the hospital, and they would just open the windows and air it out and try to let the yellow fever air out, and then mosquitoes would just come in and bite the sick yellow fever virus patients and take the yellow fever elsewhere. So making hospitals mosquito-proof was very important. They also eliminated standing water sources, the breeding grounds for mosquitoes, and they sprayed them with oil or other insect-killing chemicals. And amazingly, within, about 3 years, they eradicated yellow fever in Havana, Cuba, and in the Canal Zone, which eventually led to the successful construction of the Panama Canal. Okay, jumping to the 1930s now, Max Theiler, Nobel Prize awarded in 1951 after his work on the yellow fever virus, where he established animal models for the virus and created the vaccine that we still use today. Excuse me. And he was also a professor at Yale in the 1960s. Okay, so how did this guy in the 1930s study viruses and vaccines? Because we reasoned that you can't keep infecting people with yellow fever for research. So you really need an animal model that can be infected and has measurable pathology. And we figured out that if you inject the virus into the brains of a mouse, the mouse will die. And rhesus monkeys are also susceptible to the virus. So 2 very important animal models for this virus. And we figured out that if you can train yellow fever virus to replicate in a mouse, it'll replicate less well in a human. So what do I mean by that? So if you take yellow fever virus with its viral RNA and give it to a mouse brain and keep passaging it in mouse brains, eventually it'll acquire changes in its genetic code that make it more adapted to replicating in the mouse and less well adapted to replicating in the human. So it's kind of like training the virus for a marathon its whole life, and it'll be amazing at running long distances. It'll be amazing at being fast, but will it be able to compete in a weightlifting competition against The Rock? Probably not. So the virus is just trained now for the wrong host. So again, this process of weakening a live virus is called attenuation. So passaging the virus through the mouse, acquiring the genetic changes, mousifying the virus, essentially. And then further attenuation steps, which I'd be happy to talk about later, if anybody's interested, leads to what we now use as the yellow fever virus today. And keep in mind that this is still a live virus. It's a live viral vaccine, but it is an attenuated virus, so it is very safe. So yes, current yellow fever vaccine, there's been over 600 million doses administered. It has incredible efficacy, one of the best vaccines out there, over 90% efficacy that lasts basically your whole life, about 30 years of protection. And it is a very, very safe vaccine. So, but yellow fever itself is not just over with the vaccine. There's still 400 to 500 million people unvaccinated living in at-risk areas. And climate change is continuously influencing the range of Aedes aegypti, the vector mosquito, with a projected 8 to 13% increase by 2060 to 2080. So in conclusion, what I hope you've taken away from this talk is that RNA can be a viral intruder, which may hijack your cells and could even change human history. Medical research in the early 1900s was effective, but it was often crude, messy, and arguably unethical. And viruses are going to adapt to their host one way or another by changing their genetic code. In yellow fever virus case, it's RNA. And sometimes the virus uses that to its advantage, and sometimes we can use that to our advantage. So the yellow fever vaccines work very well, but regardless, yellow fever still remains a public health threat. So now I've talked to you about how RNA can take the form of a virus and how we can even make a vaccine out of it, a live vaccine. But Matthew is now going to take you on a journey about how we now use modern technology to use RNA as tools.
All right. So as Mikey said, you've now heard a bit about RNA in viruses. Brahmmi told you a bit about background, including Olivia, background of RNA and how we understand it and how it's can be addressed in splicing as an amazing thing, but also a concerning thing in disease. And I'm going to tell you about how we use RNA for the future and some new ways to think about RNA that you might not have before. So to do that, I first need to tell you or leave you with an analogy that I think will be important throughout the entire talk. And that's that RNA, like origami, folds to take shape. So with a piece of paper, it can act as an instruction manual of sorts. It can confer information by the written word, language, and that's very useful. But you can also take that same piece of paper, you can fold it into many different shapes. And again, that one single piece going into folded into a boat or a crane now has a new function, but it all came from that original piece of paper. And we can consider these little machines that have functions. A boat will float on water, and that's pretty awesome. And similarly, RNA can do just that. A single strand, as you saw, of RNA can fold into lots of wonderful and unique different shapes that have unique functions. But again, they just came from a single strand. And as you might see now, well, it looks like there might be multiple strands, and yeah, RNA can do that too. It can bind to itself, it can bind to DNA, and it can have these awesome structures. And this one right here is one of my favorites with the platforms. So with that, I want to show you a little bit more about how RNA does more than make protein. But I need to give you the ground framework again, just to make sure we're all on the same page, even though it was so wonderfully done before. DNA here, as we see, double-stranded, goes through a process of transcription to make RNA, and then translation to make protein. And that's all well and good. And this is not the last time you'll see this today, but there's a lot more to this picture that's not filled in yet. And that can be done with lots of different and unique RNAs that have really cool roles. So one of them is long non-coding RNA, as you see here, with lots of loops and twists here. And this turns genes on and off. And so I think this is a pretty astounding basis to now move forward with. You've heard about how we use DNA as this instruction manual and how it will code for RNA and that RNA will be a messenger. But now I've just told you, well, there's non-coding RNA that exists and that had to be transcribed from DNA to begin with. So it's already setting off on a really wonderful track of different things that RNA can do. Next, we have small nuclear RNA and small nucleolar RNA. And these help make sure that the message gets across the way that it needs to be, that it has additional features that make it recognizable and give it the A-OK to go through the rest of the steps towards translation. Next, we'll have small interfering RNA, which I'll tell you a bit about later, that can help turn genes off. We have ribosomal RNA, and lastly, tRNA, transfer RNA, which act together in concert. And these are, you can consider them little machines, as we all with the rest of these, help to make sure that the messenger RNA is translated into a protein. And so that message comes through to the RNA, the ribosomal RNA, and the protein in this picture here. And the transfer RNA also acts in that process, and we end up downstream with protein. And now I hope from this picture, what you're gathering is, wow, there's a lot of different things that RNA can do besides act as the message. And that's exactly the point. And so with that basis, now we can move a bit forward. Now you know biology can use RNA as a tool, but can we use it as a tool? And I want to give you another analogy. So if you think of a toolbox, and in our case, a molecular biology toolbox, scientists love to have different tools and resources at hand to solve and ask unique questions to help us learn more about the world and eventually act on it. And so having knowledge of these in terms of basic research and medical research gives us a lot of power toward shaping our future. And so I'm going to tell you about 3 examples now. One of them is the COVID-19 vaccine, which you are all fairly acquainted with, I imagine. And this is a tool that we now have that comes from our understanding of RNA and how we can utilize it. Small interfering RNA, which I alluded to before, which you can think about as a microphone broadcasting a message and then introduction of this interference to silence it. And lastly, gene editing, a pair of surgical tools in the form of CRISPR that will allow us to act on our genome and change the DNA to have unique features or to study it, etc., and a lot of wonderful applications that I'll get to later. But first, let's talk about the COVID-19 vaccine. So here is a diagram, a representation of what we believe SARS-CoV-2 looks like. And as you saw from Mikey's talk, this is a capsid enclosing the RNA inside. But what we want out of this, so we know this is infecting us, so it's a concern. How do we get immunity and defense against it? Well, we need to make antibodies. And with those antibodies, we can act to neutralize that SARS-CoV-2 viral particle. And that's all well and good, but how do we get antibodies? So one way that your vaccine, as you likely know now, or have heard in the news to some point, is that, my apologies, that the vaccine is mRNA based, or at least some of them are. But how does that mRNA become something in our bodies, and how does that lead to antibody production? Well, I'm going to go through that now. So here's our SARS-CoV-2 particle again. And what is a really unique thing is on the surface, there are these blue spike proteins here that help the viral particle enter into our cells and use our cells as a host, as Mikey told you about. But what is quite amazing is we know the genetic information that makes this spike protein. And because we know that information, we're able to synthesize that in a lab and put it into a vaccine. And once that vaccine is given to you, administered, that mRNA that's in the vaccine can transiently, just for a temporary amount of time, be inside of your cells. And that mRNA is then translated. As you've seen, we go from mRNA through directly, we skip the DNA at this point, we've just synthesized it, translate it into this protein, and now we have these spikes. But you might ask, why would we want to make these spike proteins? Aren't these what the viral particle uses? Well, yes, and this is where our immune system comes in. So now that we have these viral spike proteins on our cells, our immune system can recognize them as foreign and generate antibodies. And that's pretty great. That's part of the goal that we've wanted now, right? So now that we have these antibodies, the next time that you are infected by a viral intruder, specifically SARS-CoV-2, these antibodies can help neutralize those particles. And so that's a pretty amazing leap forward from just knowing the genetic code of this, what encodes this protein, we can make a vaccine where the mRNA is doing a lot of the work. And I hope what is not lost here is you've heard from Mikey how you can train a virus to be weakened against a human by giving it all this, making it run a marathon. But here, there is no marathon to run. The mRNA vaccine already gives us a huge leap forward and allows us to very rapidly move forward into getting some type of immunity, or temporary immunity at least, against SARS-CoV-2. And so that's just one example. Next is what I like to call going silent and interfering with the messenger. So again, I promise you, this is not the last time you'd see it. We have DNA being transcribed to mRNA. We can send that message out into the greater body of the cell, the cytoplasm, so the nucleus where the DNA is held, cytoplasm where a lot of other functions are held, but you can just move forward with that without explaining more. We have the ribosome, which I showed you before, that will take the message and it will read it and we'll translate it, and we can consider this gene on. So we had some type of information we wanted to look at, or we wanted to convert to protein downstream, and we did that. Great. But what if there's a mutation, or what if there's too much of the protein that's being created? Well, it would be convenient to have a means to put a stop to that. And so we have silencing or small interfering RNA that can use this Pac-Man-like structure, RNA-induced silencing complex, which the name does a lot here. And what that will do is degrade mRNA, and we'll consider that gene off. And so what's pretty astounding is in 2018, the FDA approved its first siRNA compound to be used for a pretty harmful disease, transthyretin amyloidosis, which is a mouthful, but it can lead to a lot of accumulation of these protein clumps in the body that can halt blood flow and limit nutrient flow, etc. And by using and administering this small interfering RNA, we can utilize, again, the body's machinery, its own small molecular machines, to help us get to a point where we can act on our cells and help limit how deleterious this disease can be. Okay. Lastly, a tool for genome editing, and what I hope to also impress upon you is quite astounding: CRISPR-Cas9. So in 2020, the Nobel Prize in Chemistry was awarded to Dr. Jennifer Doudna and Dr. Emmanuel Charpentier for their amazing work on and discoveries throughout the years of CRISPR-Cas9 and all the things that it can do. So what is CRISPR? As I showed you before with those genetic or those surgical tools, you can consider it genetic scissors. So here we have in blue, this blob here is a protein, and we have a RNA coupled here, helping us guide to a specific location in the DNA, which is in the lighter color here. And I'll explain more about that in a second. But essentially, this gives us the power to directly edit the DNA at a specific location, which is pretty astounding. And from that, we have a slew of downstream applications that are currently and constantly used in our society today, and will continue to be used as we move forward. And before I go into any other examples, a quick one might be, well, wouldn't it be helpful if you were gluten intolerant if we can make wheat that had less gluten in it? Well, we can, and that is something being done with CRISPR. There are certain wheat strains that we can use CRISPR on to reduce the amount of gluten content, and that is just one facet of agriculture that CRISPR is being used in amongst all these others, which I'll give you more examples later. But I want you to get a feeling of how this all works exactly. So here's that blue blob, and here is the DNA right here, and it's opened up. So this is our double strand opened up. Here is a target sequence that we want to modify. Either it has a mutation in it, or it is a great location to add in a unique or new piece of DNA that will have some type of function, either to restore function to a genome that has lost some ability to do something that we need, or to study something and we want to insert a piece of DNA to study there. So what we can do is use a guide RNA, so sgRNA here, and that will help this Cas9 system act on just this target sequence, or ideally just this target sequence. And with that, we can make many, many different guide RNAs to millions of different sequences within an endless amount of sequences within the DNA or in your genome to act upon, which gives us an amazing amount of flexibility. Further, we can then utilize the Cas9's ability to make cuts and cut at these two locations here. And what we end up with is a broken up piece of DNA. And from there, we'll use the body's machinery. So at this point, we administered some or used Cas9, we put it into the cells, and now the body is going to go through what I'd like to say is one example of a cut and paste. So this is not the only way that Cas9 can be used, but one of them, if we want to introduce new genetic information, is to also provide some type of donor template. This has additional genetic information on it, and that will be pasted into that cut section, and now we've inserted DNA. And so, again, a pretty amazing feat that we can edit the DNA at will. And here are those applications again, and two examples to hopefully tie this whole message together is that Brahmmi told you before about Hutchinson-Gilford Progeria syndrome. And in 2021, I need to get the date, right? A study was done on mice where that had this HGPS, and a CRISPR-Cas9 system was used to edit the genome, which is that one base pair that was modified to lead to all these downstream effects in HGPS. And the Cas9 system was used to modify the genome, and it ended up extending the lifespan of these mice and had a very promising result, but still lots of work needs to be done before they can be used in human patients. But one example of which they were, another alternative splicing disease, is a early onset blindness that, again, the name is eluding me, but it was used in the clinic 2 years ago, I believe, on a patient to reduce the early onset of that disease by using this Cas9 system. And just so if you're already starting to think, well, this could be quite dangerous, I don't know if I would want my genome edited or to this technology to be used in bad ways. Well, there's lots of people, especially the developers of CRISPR, who are very conscious of the ethical quandaries that the development of this technology has established in our society, and are making great efforts to ensure that its use is one, practical, two, safe, and three, is not overstepping boundaries within the scientific community and the public at large, and used for our best interests. But making sure and clarifying those bounds is something that's quite difficult and is, again, being talked about every day. And lastly, I want to leave you with this. You might be thinking, wow, these are great. You've said a lot of interesting things, but why aren't there more of them? Why aren't they done all the time? You said there's one FDA-approved siRNA. Why aren't there more? Well, so as well as we can use these tools in a research setting, or as well as we can develop them, a lot of times the delivery of them can be just as difficult. And so say we wanted to send a message, which is that RNA therapy to the pancreas. Well, sometimes that can get sent to the lungs or the intestines, and the delivery of that message can sometimes be as important as the message itself. And so clearly we don't want the lungs, we don't want the intestines, we just want the pancreas here. And that is a process in which lots of science goes into the development of all these different tools, and takes a long time to get right, to make sure that it's as safe as possible to be used. So I want to leave you with that. Science is a slow process, but sometimes it could be hastened by novel and unique tools that we can use to our advantage. And I hope today that from Brahmmi, you've learned a bit about alternative RNA splicing and how its defect can lead to HGPS. For Mikey, how viral RNA can be used in, was studied in yellow fever, a story of the science and its discovery and understanding. and a bit about how RNA is used as a tool and has a lot of wonderful and unique functions that you might not have known about, and is broadening our horizons for a future that we can utilize these tools for pretty wonderful advances in society. So with that, I'd like to thank everyone for coming, and we will take questions.
Great job, everyone. Thank you so much. Yeah, as Matt said, please feel free to type your questions in the Q&A box or in the chat, or feel free to raise your hand and I'll unmute you if you have any questions for any of our speakers today.
Someone's raising their
Yeah,
hand.
go ahead.
Is there a cure now for acute pancreatitis?
Matt, do you want to take that one? Do you know of any ways that these technologies could be used for acute pancreatitis?
I am not aware. There might be some literature on it, but I cannot speak to that exactly at that moment without looking into it.
Thank you so much.
All right, if there's no other questions, we will sign off. Thank you all very much. You've been a great audience, and we hope to be back again soon.
Thank you. Thank you. Thank you. Thank you. Looking forward to the next
Thank you.
one. Take care, guys.
Have a great night, everyone.